I cannot start my I cannot solve my device. I cannot hear anything. So
Okay. Can
Marcella,
you hear me now?
please
And now you can hear
please
me?
start your lecture. Okay, sorry.
But the rest can hear me, you think? There maybe
Yes,
sum up?
yes, we can hear you clearly.
You can hear me? Ah, okay, that's good. So that's it. But Betty, you not able to. Okay. All right, so we start, right? It's okay? You think it's okay now? Yes, okay. So I will share my screen. Good morning and good afternoon for you.
Oh, okay. Sorry, Marcella. I changed the computer. So sorry that my computer has some problem. So right now I would like to introduce you to the student. That's
Thank you.
Professor Marcela Hermoso Romero. She is our friend for such a long time. I think more than 10 years, and we have the collaboration for such a long time. And Professor first she worked in Chile, but right now she moved to University of Groningen in the Netherlands, and she has the study a lot, and she is the expert in innate immunity and the information, especially in cancer also. So for today, she will give us the example of her research about the innate immunity in cancer. Okay, sorry, Marcella.
No
So
problem, no problem,
okay,
Betty. Thank you.
okay, let's start. Thank you very much.
No, thank you so much for the introduction and the invitation to participate again in this interesting activity that you are doing in the last years, and I am very happy to participate in this amazing course. And I teach some time in Bangkok, and after the pandemic, we start doing this system online, and from Chile, as Professor Petty mentioned, and now from the Netherlands. So maybe we have less time difference now. So I want to do a brief introduction. I know that you are in this PhD program. Many of you have a lot of background in immunology, but it's not bad to do a refreshment of some information about innate immunity. Well, just to give you a brief, in the past I have some pictures from Chile, but now I want to share some pictures from the place I'm living now. This is a city in the north of Netherlands, north of Amsterdam. Netherlands is a small country, and this is a pretty picture in the evening of this city, and many other has canals and boats, and I think there's some connection with Thailand as well. And there are some agreements between the University of Groningen and Mahidol University and Sirira. So hopefully we can do some collaboration. There's a student actually from Thailand doing her PhD here in a neighbor lab, and she's doing pretty well. This is the academic building where the defenses are done. So it's a lovely building. And our this is I am in a department of gastroenterology that is part of the hospital. In this route, you can see this is the hospital. This is the main entrance, and here almost all these buildings, all this part is part of the University Medical Center. There's another campus for biology and sciences, but here we have most of the basic and translational research. So to start, we are all aware that the components of the immune systems are represented in this diagram, that we have primary and secondary organs. The limbs, the thymus and the bone marrow are primary immune organs. And importantly, secondary organs where the activation of the immune response takes place is in the secondary organs like the spleen, the lymph system, and also there are other organs spread in the surface tissues like the gut, the oral cavity, the respiratory system, and the immune cells are key in this activation or immune response. So just to remind you, all of you have a biomedical sciences training, but I will move these pictures here. All the lymphatic system is spreading all over our body, so we can have interaction of immune cells with other organs or other tissues, cell tissues all over our body. So I will move this a little bit because I prefer if you have any question before continuing, please raise your hand because sometimes I cannot see if you send a text. So I put in now. All right. So maybe raise the hand or do some, or just ask me the question. Okay. So this can be a conversation between all of us. So there are several, but it's quite nice that it's a nice group. So what activates the immune system? Well, not only a pathogen, that is the classical activation of the immune response. For example, this is a classical diagram taken from Abba's book that bacteria can activate innate immune cells. So this is super important to take in consideration which are the main immune cells participating in the activation of the immune systems. Macrophages and monocytes are very important cells in activating and detecting, for example, bacteria or other pathogens, but also are really important in the context of cancer immunology. Neutrophils are taking a big relevance also in the monitoring and patrolling of insults in our body. And natural killer cells are really trained cells to counteract the effect of viral infections as well as cancer conditions. So not only the innate immunity is related to immune cells, but also cytokines that are soluble factors that allow their conversation and interaction and activation. So there are two key cytokines that are relevant in the activation of these cells, like interferon gamma, IL-12, amongst others, of the inflammatory response, for example, TNF-alpha, IL-6, IL-1 beta. But the depletion, as is stated here in the diagram, I will put a pointer, as is highlighted here, depletion of some cells, or using blocking strategies of these key cytokines can promote the growth of bacteria in the case of an infection. And this we can also translate in the context of cancer, that if natural killer cells are not functional, or we don't have a proper production of these cytokines, the tumor cells can grow in an exponential way, similar to what we see here. So in some way, the innate immunity, as is shown here, is the first period that is taking place in the first days of an infection or an immune reaction to control this situation in the case of infection, but they are relevant to activate a more long-term immunity, that is the adaptive immunity that we can see here after 5-7 days. This other arm of the immune system is activated, but requires some cellular and molecular events to take place before T cells and B cells, that are the main representative of this immunity, are active to eradicate, for example, the infection or pathogenic bacteria, or in the case of a aberrant cell or a tumor cell. And again, if we deplete experimentally the T cells, for example, in cells in animals that are deficient in T cells, or in the case of an tumor cancer condition, effector T cells that CD4 and CD8, mainly cytotoxic T cells like CD8, are silenced or are decreased. The expansion of this situation is increased, and the control of the infection or the tumorigenicity is lost, so it's not potentially to eradicate and go back to the homeostatic condition. There's any question about this cartoon or diagram? Please, you let me know, okay? So this is, as I said, as a way to represent how the two arms of the innate immune system control an infection, but also how they can control another insult like a tumoral condition. So if we go back to the innate immunity, more than almost 15 years ago, in 2011, 3 researchers from different countries, from France, from Canada, and from the US, they got the Nobel Prize in Physiology and Medicine because they were able to demonstrate the relevance of the innate immunity. So you can see that it's not that long, the recognition of this arm of the immune system. The innate immunity was mostly considered like a phagocytic activity, but not very specific. However, you might have some information that the innate immunity, as we describe, is very rapid, can halt the infection, has no memory. However, now there are some debate on that. And the adaptive immunity is a slower activating response, but can clear, in this case, an infection, as we were talking, and has a memory. However, you can see that this individual that is confronted to this pathogen, as is shown in this first part of the diagram, there's like a protection first from the innate immunity, that all our barriers, for example, the skin, the gastrointestinal tract, and as we will see, the lungs or other surface tissues are representing the innate immunity. You see all this green highlighting in the surface of the body. However, the innate immunity is spread all over our tissue thanks to the effect of the lymphoid organs that are distributed all over our body. So how the innate immunity is activated? Well, we now know through the research of these 3 investigators that there are pathogen recognition receptors, like you are quite familiar with toll-like receptors. This is one family of receptors, but you know that there are other ones, that some of them are distributed in the cell membranes, but also they can be soluble receptors. And the main cells that are expressing those receptors, these are the dendritic cells, in terms that they can connect the innate to the adaptive immunity, because remember, the adaptive immunity is mainly tailored by the T lymphocytes. So the dendritic cells are key cells that can migrate into the lymph nodes and activate the naive T cells presenting an antigen. These are antigen-presenting cells, but are professional antigen-presenting cells. So to summarize what Jen Wei and if you can see, even in 1998, almost 25 years, they demonstrate that he demonstrates through different evidences, and this is a nice paper, that the innate immunity is not unspecific, because it has contained receptors, as we saw, that are fixed in the genome. They can recognize conserved molecular patterns that are called pathogen recognition receptors, and they can discriminate the self from non-self in a very selected period, and is quite conserved in the evolution. So you have innate immune response even in amoebas and very ancient organisms. And the adaptive immunity, you see some molecular representation of the adaptive immunity in fish, for example, or however, in C. elegans or another organisms, more evolutionary ancestry organisms, only the innate immunity is present. So that means that is quite relevant for the survival of an organism. And the action time is immediate, and the response is mediated by cytokines and chemokines. So these are 2 components that we will mention along the lecture quite often. So briefly, maybe you are familiar with these diagrams, but the innate immunity, as I said, are mainly in our barriers, epithelial barriers, for example. So we describe phagocytes like monocytes and monocytes or macrophages, dendritic cells that are also derived from monocytes, the natural killer cells, and here are also important the neutrophils. And in these epithelial are mechanic barriers, such as the skin, the mucosa, they have cilia, and chemical barriers are produced or present in these surfaces, like the production of mucus, the change of pH, and enzymes that can be important as bactericidal molecules together with defensins, complement, interferons, and lysozymes that are locally produced by specialized cells in the epithelial barriers, not only in the gastrointestinal tract, but also in the airways or other tissues. Recently, and I will try to summarize a little bit in the class, that other biological factors are the gut microbiota and the skin microbiota are important as contributors of this innate immunity. In the adaptive, we see that it requires more time, because the B and T cells, they require to have a maturation and proliferation. First, recognize in the lymph nodes the antigen from a bacteria or from a tumor cell, and then activate this clone of cells and proliferate and go back to the organ to recognize, for example, the antigen or present in the tumor or in the infection site. And the effector molecules are the antibodies. They are super important in viral infection, bacterial infection, as well as in tumor immunology. And nowadays, you know that there are some therapies that are based on these kind of molecular structures that are biological therapy or based on antibodies towards some important molecules. So in the case of the T cells, there are two effector T cells that we know as CD4 or CD8 or cytotoxic. That the cytotoxic T cells, they have the similar or comparable effector function as the natural killer cells. So these are, I brought these images because not all of you are pathologists, but some of you maybe have some interaction and have some insights, but these are lovely images of the respiratory system and the skin and the gut. So these are the first barrier against an infection. And sometimes during infection, we have inflammation, right? And this inflammation requires a resolution process that if it takes too long, it can get some errors, and these cells can proliferate aberrantly and generate aberrant cells in these different organs. So also in the these physical barriers are, then we will integrate all of that, but there are key components, as we mentioned, that are the defenses of or antimicrobial peptides that are produced by specialized cells called Paneth cells in the gut, for example. Also in the mouth, we have enzymes and cells that are producing these antimicrobial peptides, as well as the directional flow in the stomach, and the pH and digestive enzymes are relevant as physical and chemical barriers. And in the large intestine is more relevant the commensal flora, the process of tolerance that are mainly represented by T subpopulation of T cells called T regulatory cells that balance the function of the effector T cells like CD4 or CD8 T cells, and resident macrophages that also contribute to the tolerance. And in the case of tumor conditions, they can become allies of these tumor cells, making a beneficial microenvironment for the cells to proliferate. And these are called macrophage-associated, tumor-associated macrophages, that they have a kind of tolerogenic phenotype. So there's a balance, of course, to keep a non-inflammatory mucosa, however, being able to respond in terms of phagocytosis and elimination of a bacteria or pathogen. And in the airways, not only there are present resident macrophages, but also there are ciliary cells or mucus. And to represent that, in this diagram that maybe I will share later the class, there you can see that is in this tissue the production of this mucus layer that is quite prominent and is a physical barrier, but also contribute to the energy source for the microbiota that is represented by these elements that are in the top part. And you see that some are in between the mucosa. So these beneficial bacteria can be get their energy source from the glycoproteins present conforming this mucus layer that are mucins mainly in all these tissues. And in the gut and in the for example in the lung, there are production of immunoglobulins that are relevant also to keep a balance between beneficial and pathogenic bacteria. And these are able to be, in the case of the gut, the main immunoglobulin. You remember which is the main immunoglobulin in the intestinal surfaces? Somebody remember which is the main immunoglobulin? Is the IgA that is relevant in controlling this effect. And it has been shown that for example, in colorectal cancer, the production of immunoglobulin A is reduced, as well as in inflammatory conditions. For example, in Crohn's disease, the secretion of and the proliferation and the differentiation of plasma cells is very low. And this makes sense because the production of TGF-beta that is relevant for the switch class from IgM to IgG is altered production. However, in tumor conditions, even you have a microenvironment rich in TGF-beta, there can be a shift toward fibrosis. So there's a loss of balance in the functions in these pathogenic conditions. So having said that, this is a nice diagram from a review from Pastorelli that they show how other cells, these are the secretory cells, maybe you remember the goblet cells are important in the production of these mucins that are keep away from the luminant antigens, put a physical barrier. However, there are other alarmins that we will talk later in the class. For example, we were working with Professor Petit Touajid and Chanitra Touajid, that is interleukin 33. That is relevant in alerting the immune system that there's a danger, for example, damage or necrosis of the tissue, because these, like other alarmins, are secreted upon tissue damage. However, they also can lead to fibrosis and collagen deposition. So for example, in pathogenic conditions, this can be a marker of pathogenicity. So the mucin is the main glycoprotein and requires these glycosylations that are important in not only in the capture of the water, but also keeping the moisture of this mucous shell, but also promoting the secretion. You might remember that glycosylation is important for proteins that are secreted to be extracellularly placed. And during inflammation, there's reticulum stress. Maybe you've heard of this condition that it happens in inflammatory and pathogenic situations. So the post-translational modification of molecules such as glycosylation is aberrant. So many of these proteins are kept intracellularly. So in a pathogenic tissue, you will see that the mucus layer can be reduced. So this is something interesting to consider when we talk about inflammation and that these ER stress processes are taking place. So these are other functions that when I mentioned, and here just to show how these these cells are exhibited in a tissue section. They look quite nice, these panel cells. And antimicrobial proteins, not only defenses, but also enzymes like lysozyme, and lactoferrin, that complex iron that can be relevant for bacteria metabolism. And the natural antibodies are IgM and IgA that are relevant to block and neutralize toxins in these tissues. So this is a cool image that was taken with a microscopy, that you can see a bacteria like the Escherichia coli in the presence or absence of defenses. How relevant are these innate immune molecules to take a control of a bacterial infection, for example. So these defenses are not only in the epithelial layers. This is a table that show that epithelial cells are the main source of these defenses. However, mononuclear cells, macrophages, and dendritic cells also are important in the production of these defenses, and they can be induced by inflammation, for example, through interferon or other cytokines, as well as pathogen recognition receptors like LPS, peptidoglycans, and so on. So the neutrophils are key cells, and I will give a brief overview of the cells of the innate immunity. And the neutrophils are relevant, secreting antimicrobial proteins. And now it's not only relevant in the production of these antimicrobial proteins and enzymes. You can see that they harbor several and different type of granules, but also they are producing extracellular or nets that trap bacteria and neutrophils. And nowadays, this is a very hot topic, the neutrophil traps or the nets, because they not only can be a reaction to protect against bacteria or other pathogenic infection, but also nowadays are relevant to activate the immune response, because the nets, they have also proteins in their composition. The nets are traps of DNA, but also proteins, so they can activate and recruit antibodies to these autoantigens and set an immune response, activating, for example, macrophages, because you remember that immunoglobulins, they are important in neutralizing and recognizing antigens. However, they have another part in their in their molecule that they can activate the innate immunity, for example, through Fc receptors and macrophages or other immune cells. Sorry, I will drink a little water. So this is an overview of how the antimicrobial peptides can not only function as a bactericidal molecule, as we saw in this nice microscopic image of bacteria. However, they can also be chemotactic molecules, and you can see that many of these defenses can bring to the site of battle site dendritic cells, not only innate immune cells like dendritic cells, macrophages, and neutrophils, as well as mast cells that I haven't mentioned much up today. They are orchestrator of the innate immunity because they can degranulate as they are polymorphonuclear cells like the neutrophils. But also these defenses can recruit T cells from the periphery into the tissue. So in a tumor microenvironment, the production of defenses is reduced. So also is a mechanism to damp the activation of the immune response. So as I said, the dendritic cells are key cells bridging the innate and the adaptive immune system. These cells, they are called dendritic cells because they have these prolongations or dendrites. This is a staining of a dendritic cells, a dendritic cell, and they are able to recognize pump through these receptors described by the Bruce Boutler, for example, one of the researchers recognized for the medicine Nobel Prize. And these receptors, you might heard many times, they activate signaling pathway leading to the differentiation of these cells, which are the main molecules activated through this signaling pathway, mainly NF-kappa B or other transcription factor, or like that leading to the production of chemokines, cytokines that are soluble factors. However, these cells also are differentiated into antigen-presenting cells, because before the recognition, they are not expressing, for example, molecules involved in this antigen presentation, like the HLA or co-stimulatory molecules, like CD80, CD86. And why they are important, this presentation? Because in the lymph nodes, they can activate naive T cells, depending on the antigen presentation. It can be CD4 or CD8. Remember that through HLA1 or HLA2. And this cellular immunity is relevant to for cytotoxic effect or activate other cells of the innate immunity, for example, macrophages, but also to activate the other arm of the adaptive immunity, that is the humoral immunity represented by the B cells, so antibodies can be produced. So these elements are these bridging mechanisms towards the activation. So I want to now put in context how these immune cells that I described, and do you have any question about the introduction of the innate immunity and how activate the adaptive immunity? Do you have any question or comment? Some of you? No? So I take as a no, that all of you can hear, right? I think, so how these immune cells eliminate the pathogen and abnormal cells? That is our main goal for this class. And which are these cells? So if we have aberrant cells or cancer cells, we have this first line of defense that we talk about, the NK cells. And if this is a target cells of a cancer cell, for example, this T cell can destroy or through apoptotic mechanisms these tumor cells. So let's see, I will show briefly this. I don't know if I can open, but I have open in my here. Give me a second. Oh. Give me a second, because I want to, I don't know how to do it. I have opened the, I think it's open here.
Inside all of it lurks a series.
Let's no, silence the. So we this is, I think is quite nice. So share this image. So let's go. So it's 2 minutes. We can see that. Can you hear me, right? So this is a tumor cells, and the cytotoxic T cells or NK cells, they polarize and they can destroy the tumor cells. You can see that how the red cells that are tumor cells die with the fluorochrome, they can eliminate. And we have, as I say here, many millions of T cells that can recognize infected cells, for example, through viral infections. And they can also recognize tumor cells. So when the cytotoxic T cells find the cancer cell, the membrane protrusion, you can see this is very nice image. This is made on in vitro. They can kill the target using poisoning proteins visible here in red, that are the cytotoxic granules, perforin and granzyme. So they do a polarization. You can see like a very fine interaction. And all these granules, this is quite impressive image. It was taken a few years ago, but you can see how all these granules, they polarize towards the contact to the target cells. So here, this is very nice. I will share with you. This is from the University of Cambridge. So we'll close that and go back to our. So do you have any comment? I don't know. I am. Let me see if I am. Wait. I don't know if I am. No, I have to share again. Sorry. So, any comment or can you hear me? Could you hear the video I presented?
Yes, ma'am.
Oh, perfect. Great. So we go back. Do you have any comment on the video? Have you seen something like that before? So here is, I put just the last figure, but here is the link. You can check when I send you the class, you can see again in detail and discuss with your colleagues. But these are examples of T cells, the CD8 T cells, as well as the natural killer cell, how they can protrude and polarize the production of their granules of perforin and granzyme, so they can destroy the target cell. So inflammation is a quite important aspect. So the antigen-presenting cells through this TLR are relevant to detect pathogenic, like in this case of this infected tissue, but also tumorogenic antigens, and activate intracellular pathway. However, in tumor conditions, this inflammation is quite reduced. And I will show you some evidences that after chronic inflammatory disease, you can have a aberrant cell generation. But this, physiologically talking, inflammation is a beneficial response to eliminate an insult, right? For example, a pathogen. And these are the characteristics you maybe probably saw in different ways of representing that the heat, the redness, the swelling, the pain, and the loss of function are the main hallmarks of the chronic inflammation. So how we can link the inflammation and cancer? I am, as Professor Petty was mentioning, I am working for several years in gastrointestinal conditions like Crohn's disease and ulcerative colitis that unfortunately are rising all over the world. Even there are some reports that in Asia, that was one condition that was not relevantly prevalent nowadays. The Crohn's disease and ulcerative colitis are more prevalent, and there's an increase in the incidence. These are numbers in the US. However, they manifest similarly that they have some clinical heterogeneous phenotypes. They have multiple factors involved, so it's quite complex. Like many inflammatory conditions, autoimmune diseases are multifactorial as well, and we can categorize these kind of diseases like autoinflammation, autoimmunity. However, there are not particularly autoantigens involved here. They have no cure, and most of the therapy, the patients are refractory in between 30–40%. So many of them require surgery, and they are chronic diseases. So they have the risk of developing colorectal cancer. So this is an important study made for few years ago. However, they demonstrate that after 10, 20, or 30 years of having a patient with ulcerative colitis, Crohn's disease, or pancolitis, that is like extensive colitis, maybe now is changed the terminology, but 10 years ago around was the terminology used. You can see that there's a cumulative probability of developing colorectal cancer. And these are the analysis of many articles of different cohorts of patients. And you can see that after 10 years of diagnosis, you see an exponential increase of the probability of developing colorectal cancer. So we have here, you maybe heard of sporadic colorectal cancer, and the colitis-associated colorectal cancer. They have similarities in their histopathological representation, that you have dysplasia, adenoma, and carcinoma. However, the factors involved are quite different. One of the main factors in the colitis-associated colorectal cancer is inflammation. That is something that you don't see in the sporadic colorectal cancer, right? That is more a genetic related condition. So in this, in the upper part that we are talking now, the inflammation that I was talking before, inflammation is a physiological condition to eliminate a pathogen, for example, or an insult that is a toxin. However, if this process is chronic, this epithelial repair can have some errors because of this repetitive process, and can, due to this damage and regeneration, can become some errors. And there are some aberrant cells, as you can see here. And this, in the dysplasia portion, you have a lot of inflammatory cytokines that from the innate and the adaptive immunity. For example, interleukin-17 is mostly represented for Th17 cells, T helper 17 cells that are classically observed in autoimmune diseases and in these inflammatory bowel diseases like Crohn and colitis. TNF-alpha is a hallmark of inflammation, the same as IL-6, and the STAT3 pathway that is activated by IL-6, among other cytokines that have been described in many tumor conditions, for example, cholangiocarcinoma, that Professor Petty and Chanitra Tuaji, they work extensively in this condition, and IL-6 is increased in the initial part of the tumorigenicity. However, this is a very ubiquitous cytokine that is important in the, physiologically speaking, in activating B-cell proliferation is quite important. However, if this pathway through the STAT3 pathway, JAK-STAT pathway, is chronically activated, can lead tissue damage and fibrosis, for example. So this is the characteristic of dysplasia. And if you see high-grade dysplasia, also inflammation is represented. However, other pathways now are activated, like KRAS. And a carcinoma is, there's other pathways involved. However, if you see during the first 30 years of chronic inflammation, all these potentially these processes can be taking place. So as many other inflammatory conditions, there are relevant aspects to take in place that all, you see, all the immune cells are involved in this kind of diseases. So the immune system is super important with the two arms, the innate and adaptive, and also the stroma cells are quite relevant because there's a strong connection between the stroma cells and the immune cells. And you see that in the tumor microenvironment, fibroblasts take a pivotal role. However, the genetic susceptibility is important in some patients, and the environment. And the gut microbiota is part of this environment. And we said that gut environment can be the diet. And I share with Professor Petty to share with you some papers, recent papers from some colleagues that we show that the change in the diet can shape the kind of microbiota. So this is something that you as medical students or PhD student in biomedical sciences has to take in consideration how the diet can shape the microenvironment and the microbiota, and this can impact in the immune system because we focus for many years, for many years, how the genetic variants can shape IBD and other diseases. And this paper of Nature Genetics from 10 years ago, actually, you can see in the Crohn's disease and ulcerative colitis, this was a very nice article that they compare European population and East Asian population, which are the main genetic risk variants in IBD, right? 10 years ago, there was an increase in the incidence. And you can see that in contrast to the European population, that pathogen recognition receptors were described like not two receptors, intracellular receptors, or here is the interleukin-33 receptor is important. You can see that TNF and receptors and death domain receptors are relevant in the Asian population as genetic variants. And also for ulcerative colitis, that is considered more autoimmune disease because there's some variants of HLA, DR or the Q that are involved, but in different proportion. It's quite more strong in the Asian population. So this is a paper that you can review if you are interested, mostly speaking how a genetic risk variant can shape or make susceptible to an individual to develop a disease. However, other risk factors are important. For example, for colorectal cancer, I was as Petty mentioned, in South America, the incidence and mortality is increasing. These are data from 10 years ago or more, 2012. And there's a paper in 2010, but nowadays there are more updated information that place the risk in the second and third, and the mortality the third place. But you can see that the genetic is not that relevant. What is the main risk factor? The diet and the smoking and alcohol consumption, and also the family history. We saw that the sporadic colorectal cancer, there's some genetic influence, but it's the last factor involved, the IBD as long time development, and the metabolic diseases or obesity. So this is a paper published with Professor Petty and Chanitra Dwajit some years ago, that we tried to define with some talented young investigators from Chile and Thailand, that which are the main factors participating in the process. And we were like also studying this metadata, that how IBD can shape the developing of colorectal cancer, but also we were interested in cholangiocarcinoma. That is, there's a high incidence in Thailand, right? So an injury and an infection can promote the disruption of this epithelial barrier, and inflammation can promote the repair of this damaged epithelial barrier. But if this process is prolonged, this you can see that what are the main contributors. And you see that there are a lot of immune response elements, from the innate and the adaptive immunity. You have neutrophils that we described at the beginning, macrophages that in the barriers are patrolling the toxins or toxin on infection, but there's a shift from M1 that are more reactive, more phagocytic, more effector cells, towards M2 macrophages that are more important in the production of interleukin-10 and promoting proliferation of neighboring cells. What else we have here? We have Th17 cells that are important in the permeability of and promote the angiogenesis. And stroma cell, like the fibroblasts, are present as structural cells. But look what happened in the next step. Now we have the fibroblasts are more active, and they are expressing markers of activation like alpha SMA, smooth muscle action, and that they can produce TGF-beta. And TGF-beta, together with IL-10, produced by immune cells, they can shape a tumor microenvironment favorable for these cells to proliferate. And together with these cytokines, like TNF and IL-6, this proliferation takes place. And then we have that this fibroblast differentiating cancer-associated fibroblasts, that there was several papers of Professor Dwajit that demonstrate some even markers for cholangiocarcinoma and other tumor conditions, that they produce other soluble markers like periostin and other soluble marker chemokines and cytokines that are biomarkers and potential targets of therapy. And the tumor-associated macrophage are like a variant of these M2 macrophages, because they express, if you can compare, almost the same soluble factors like metalloproteinases and anti-inflammatory or tolerogenic cytokines. So they are not able to eradicate. So they are mostly supporting this tumor condition. So there's a yin and yang. This is a balance, right, between the function of the innate immunity, the inflammation in the onset and resolution, and also in the cancer, if they can be eradicated, or immune escape. Immune escape means that they can avoid or evade the immune response, not only the innate, but also the adaptive immunity. And here takes place the immunotherapy that we use all this information as developing prognostic biomarkers. So there can be immunotherapy for, but mostly a personalized immunotherapy because it's so complex. People don't have exactly, depending on the ethnicity, the microenvironment, they are immune and adapted, and innate immune response are not exactly the same. So this is a paper from 2-3 years ago, Nature Genetics, that for example, just for colorectal cancer, and this is an example, we are not going to analyze because this is not a genetic or single cell class, but just to show you what is going on in the research field of this kind of diseases, that through using single cell and bulk transcriptome sequencing, we can identify epithelial cells status that can important to generate tools as biomarkers or of prognostic of the patient, and eventually some signaling pathway that can lead to novel immunotherapies. Because even, as I said, for inflammatory conditions in cancer pathologies, patients also have some resistance or they don't respond to the therapies. So this is more complex than using one or two. So are you all familiar, or what is the bulk transcriptome, single cell transcriptome sequencing? Anyone want to give some comment or? So this technique, are you all here in the class, right? So this technique is very useful nowadays because we can see bulk means like you put the whole tissue, but we are now aware that in a tumor condition or even in a healthy tissue, we have a huge diversity of cells in different status. So with the bulk transcriptome, it's like you put in a blender fruit and you get the mix of all the genes you have in this tissue, right? But you cannot ascribe to which kind of cells are these involved. So with a single cell transcriptomic, you can identify the major cell types. So through this single cell, you identify a specific subtypes, in this case of epithelial cells, but also you can see a single cell of a tissue that has immune cells. So you also can see the immune cell population using a specific gene markers previously described in the literature that, for example, are Paneth cells or secretory cells, as we saw in the column, right? And then you mark as different groups of cells. Some maybe acquire some tumorigenesis or proliferative proliferation genes. So you can build heat maps to see in a cohort of patients, for example, with healthy tissue or tumors, how these genes are distributed and how these epithelial markers are distributed. So here, just to give an example, in this bulk transcriptome dataset, they describe this they have a lot of data sets, and they compare with the a single cell database that they analyze, and they can integrate all these analyses to see, for example, nowadays you can do interaction analysis, all from artificial intelligence, how these signaling pathways are activated, where are the sender cells and the receiver cells. They are interesting studies that you can then prove in the laboratory if these processes are actually taking place. So what they found in this study, to go briefly, is that there are some epithelial tumor cell states that can have some relevance in activated immune responses or immune signature, that there are a group of genes that are important. So here is one of these colorectal cancer cluster, and this is the other one more related to mutants, BRAF mutants, KRAS, and also other cell type signature, for example, of the endothelium or in the stromal cell compartment, but also some innate immune signatures that are related to dendritic cells or NK cells. So I want to take you more into the how molecularly speaking, these inflammatory pathways can be taking place. So from 2022, we go to 2006 to some seminal studies that were done in patients with these inflammatory conditions. And then we want to see how these signaling pathways can be relevant in the activation of tumorigenesis. So the TLRs are these innate immune sensors, and how they were related to IBD. And this is a study, as I said, of almost 20 years ago or more. But it's quite interesting that they found that some nucleotide variants or single nucleotide polymorphisms in these toll-like receptors were associated with different kind of ulcerative colitis and Crohn's disease. This is Crohn's disease, and the different phenotypes. And you can see that the risk genetic variant are more related in this case with the more severe colitis, like the pancolitis. And around that period, a group in Harvard, they show in 2004, this is a study done in animal models that deficient in some of these receptors, they check the survival and the weight change. That is a feature of patients with these inflammatory conditions. And they subjected these animal model of colitis that is using a chemical that make ulcers in the epithelial layer. So it's a very acute and aggressive model. However, it's extensively used, and nowadays still is used, believe me. And you remember that this toll-like receptor have a common adapter that is NYD88, and you can see that it's more severe, the survival of these deficient animals when they lack the signaling pathway of the TLR. All of you can follow me, because I don't want to, or you need to, you need me to explain the signaling pathway? Can you comment, please? Are you familiar with that?
Yes,
Betty, you think? Yes? Are you all familiar? Yeah, because if you are not familiar, please let me know, because I continue and I think I assume that you all understand, right? But please, if there's something that you want to comment or you need to clarify, please let me know. So this animal model, they see that they recapitulate other characteristic of the disease in humans, that there's a blood in the stool, and as you can see that there's an increase in the colonic score of blood content, and there's inflammation as well. There are production of ulceration and erosion, and other epithelial injury, and infiltrating leukocytes. However, there are not much difference between the deficient mice of MYD88 of the wild type after 5 days of this colitis exposure model. So they compare, and this is very nice. I think it's a very classical paper. It's in Nature. So also you can download it if you like, or we can give you the access, but it's publicly available. When the animal, the wild type animal, they subjected to antibiotics, look what happened with the cytokine production. It's exactly the same, or it's in the same level of the deficient animal. So they cannot produce IL-6, TNF. And we were talking that these are inflammatory and cytokines that are not beneficial, right? But the inflammation here, there's an evidence that the inflammation is quite relevant to take place, because it's important for the healing of the mucosa in the case of the colon. So what happened in an individual is exposed to antibiotics, they don't produce TNF or cytokines. And most importantly, this cytokine growth factor, KC1, that is important for the epithelial cells to regenerate. So if you have an aberrant TLR signaling or antibiotic exposure for a long time, this is what happened, not only in the mice, but also in other mammals, like the human. And the production of IL-6, you see, is very poor compared to the wild type in time. And also the keratinocyte growth factor that is important for the epithelial cell regeneration. So, well, we can conclude that the innate immune signal are important and protector to restore the tissue damage and epithelial integrity. So what happened in this you see is 5 years after that previous paper, they took the same conceptual idea, but they generate a carcinogenic mice model using DSS in a chronic exposure, so different period of exposure to dextransulfate, and AOM promote the tumorogenesis. This is a mutagenic component, and they measure the similar scores, the same scores at the end of the study, that is after 2 months, right? 60 days. So they check the bleeding score. So we can say that this is a colorectal cancer mice model, right? So they measure and they compare to the NYD88 deficient mice, similar to the previous one, but in that one was acute colitis. This is chronic plus the tumorogenic compound. So the bleeding score is significantly increased, diarrhea score. What else? The colon length is other characteristic that I didn't show in the previous paper, but they also analyzed that there's, even in humans, there's a shortening of the colon as a compression of the tissue. Polyp numbers, you can see these are increased in the NYD88. Inflammation score is also significantly increased. The ulcers, also much higher number. Metaplasia is present not in the wild type, but in the MYD88. And what we see here on the right panel, that after the administration of AOM, there's a amount of polyps as well. This is after, here they show after 60 days, but here they show the time course almost, how much is 6 months, right? There's the survival is quite dramatic. Mostly 90% of the wild type animals survive, but the other ones, there's a quite important loss. Then you see the histological characteristic. They are completely different. You have, I'm not a pathologist, but you can see a quite dramatic difference in the epithelial layer and the mucus layer, and also the appearance of adenocarcinoma, right? So the innate immune signal regulate the inflammation, but also carcinogenesis in the colon, right? So there are many pathways. I built this cartoon long time ago. There are many pathways that regulate it, so like receptors. Here we have some examples of in humans of single nucleotide variants of the extracellular domain or the intracellular domain, but also we see that there are some splice variants of NYD88, so short variants that they cannot integrate the intracellular domain of the TLR with the signaling pathway downstream. Also, we have some, this is a kinase associated to the interleukin-1 receptor, and they are there are different variants. For example, the IRAC-M silence the signaling pathway. This is has no catalytic activity. Also, there are some expression of endogenous substrates like TOLIP that also they block the signaling pathway, as this kinase phosphorylate this endogenous substrate and block the activation of NF-kappa B related genes. So, and there are many more. These are some examples. So why I'm giving you this information? Because a paper of 2006, they also give some evidence that this variant of IRAC-M protects against colitis, but can enhance tumor growth. So this is quite interesting paper because they they demonstrate that what is the impact of the absence of IRAC-M, like a deficient animal model, as we saw previously, that they can change the body weight change or loss in a colitis model, but also they can see what are the impact on a proliferation signaling pathway, for example, the beta-catenin. So they said that when IRAC-M is absent, there are some inflammatory scores, increase of CD45 cells that are myeloid cells. However, when they do a chronic model, they see some protection of tumor growth. For example, they have more inflammation, but they can also, when they are deficient in this kinase, but they have less incidence of tumor. So it's like a functional or molecule, right? And other alarm, we talk about alarmins, and there are many more, many molecules that are all endogenous molecules, some related to the there are nuclear proteins like HMGB1 that are associated with the recognition by toll-like receptors, heat shock protein, calprotectin, some defensins and galectins, and this is a modification, a chart I made modifying a paper from some years ago in which we can see that some molecules are passively released and other ones require the active secretion through several factors. And I want to call your attention that some of these alarmins or danger-associated molecular patterns like HMGB1 or calprotectin or interleukin-33 are important in the tissue regeneration. So these alarmins are endogenous. They are produced after necrosis and can recruit some, I will put the laser, that can recruit and activate immune cells from the innate and adaptive immunity, promote homeostasis and tissue healing after direct or secondary inflammatory effects. So I will give you an example of one of these alarmins is IL-33 that were described in several inflammatory conditions, but also in cancer. So IBD, we described some years ago, but also now there are some examples of the relevance of these alarmins in cancer. So these alarmins can be released after damage, as we saw occur from necrosis, for example, in these animal models of colitis or colorectal cancer. They can release this alarmin from the epithelial or the stromal cells, and also in the endothelial cells that were initially described, and can activate a plethora of immune cells from the innate and adaptive immune cells, and can have a, I prefer this. You can see, right? Because with the laser, I cannot move the presentation, but they can, promote the differentiation into more tolerogenic phenotype, more regeneration, because that's the role of this alarmin, to promote the mucosa healing or the tissue healing. However, if this is perpetuated in the time, it can lead to aberrant regeneration of the mucosa. So we described some years ago, characterized in IBD patients, but also we characterize the response to therapy that are used like corticoids, and corticoids can promote the expression of one of the variants of the receptor, a soluble variant of the receptor that is a severity and activity biomarker. And then with the we characterize the role of this pathway, IL-33-ST2, in colorectal cancer. And we found that IL-33 is related to stromal tumor-associated macrophages because we stain the presence of the alarmin and the receptor that is named ST2. And we found that there is a significant correlation with the presence of macrophages through CD68 and with the phenotypic characteristic of tumor-associated macrophages like CD163, that is also an M2 marker, but if we found in not only in the tissue, but also in the metastatic fibrotic lymph node, like we can see here. This is a staining of the lymph node that we can see that IL-33 and ST2 is present in this tissue. So we think that this signaling pathway is involved in the colorectal cancer, because we also did some studies in epithelial colorectal cancer cells, epithelial cells like epithelial cell line HT29 that we saw EMT, epithelial mesenchymal transition. And also we found that IL-33 induced desmoplasia in cancer-associated fibroblasts. We think that IL-33, that is the purple plot, circles, are secreted by the epithelial or tumor cells, and they can impact in the macrophages, producing some profibrotic factors, and together with IL-33, they can promote, that is known that IL-33 together with IL-13, activate IL-13, induce fibroblast activation. And desmoplasia, as you might know, that is a metastasis-related factor in colorectal cancer, like together with tumor budding and lymph node metastasis. We found that the epithelial tumor content of IL-33 has an association with the stromal content, so of the receptor. So we describe a model in which these are all the components of the tumor microenvironment that are directed or shaped by this example of an alarmin that can be involved in chronic inflammation and tumorigenesis. So also there's a interesting study in colorectal cancer published by the group of Professor Chanitra and Petit-Touagit, that they found that IL-33 also in colorectal cholangiocarcinoma, when it's expressed in cancer cells and cancer-associated fibroblasts, can be correlated here with a good prognosis and suppressed migration of cells. So here is an example of how IL-33 is expressed in cancer-associated fibroblasts and cancer cells, and how they, using a knockout of cells compared to the parental cells that do express IL-33, the wound healing and migration of fibroblasts is impaired when they are present with lack IL-33. So I want to briefly introduce other factors. We have time. If you have questions, please you raise the hand, but I want to show also other factors that are mentioned that influence the inflammation of this surface barrier, like the gut, and can contribute to the tumorigenesis, that is the microbiota. And the microbiota can be shaped by the diet, but also by host factors and the genetic susceptibility, immune system components, as we talked briefly, and the pressure, selective pressure of the use of breastfeeding or antibiotics. So we see that it's clear that the microbiome can shape the immune system. And you can see that animals that are deficient in or exposed to antibiotics, sorry, they have anatomical and histological changes, like we didn't talk in detail, but follicle organization of the secondary lymph nodes in the gut mucosa are called the Peyer patches, that are, and also isolated lymphoid follicles are poorly developed and less number the spleen is also reduced in size. And immunologically speaking, there are a reduced production of immunoglobulins, as I said before, remember, also not locally, but also in the periphery. The number and activation of T cells are significant, dramatically reduced. Also the cytotoxic activity, the homing of lymphocytes to the tissue, that means the influx and the arrival of these cells to the tissue is impaired or very affected. And functionality, you can use the microbiota deficient animals or exposed to antibiotics. You see that an increased susceptibility to infections and reduced ability to induce the tolerance in the mucosa. So the cells are mostly all the immune cells that can be relevant in shaping the intestinal microbiome. And we see that the same as pathogenic bacteria, the microbiota are not only bacteria, but also viruses and fungi. And they have these pathogen-associated molecular patterns. So we call commensal flora-associated molecular patterns, or CAMs, that just as the pathogen associated molecular pathogen, they can bind to these receptors and they can activate signaling pathways. So they are super important in shaping our mucosal immune response. How do they do that? So this is a very classic experiment done in animal models, so that they check where are the immunohomeostasis, which is the tissue, and they treat these animals with antibiotics to see which are the content of the T regulatory cells that are important in controlling the tissue tolerance, right? And balance the function of the effector CD4 and CD8 T cells that are also present in the mucosa. So see what happened in the colon. The number of FOXP3 positive CD4 cells, FOXP3 is a transcription factor of T regulatory cells, is the higher levels are in the colon, lower levels in the small intestine. And you can see that in another tissues like a thymus, spleen, liver, lung, they are represented, but in much less proportion than in the colon. You see, this is FOXP3. This is, are you all familiar with cytometry plots? Yes? Hello? Are you all familiar?
Yes.
All right?
Yes, sir.
Yes. So you agree with me that the colon lamina propria express the highest level of T regulatory cells that are super important in controlling the balance and state of no inflammation, right? In homeostatic condition. Look what happened when these animals are exposed to an antibiotic, right? What happened in the colon is dramatically reduce the number of FOXP3 positive cells. So see how antibiotic exposure, and we saw the impacts of the reduction of the Tregs. What happened if we don't have Tregs? There's an increase of the counterpart that are the effector T cells. We saw in these tables that other pathways are impaired, right? So it's important that the colon had a balanced Tregs number, so we have a balanced and tolerant mucosa. So the oral inoculation of, also this is another experiment. It's in a science paper of almost 15 years ago, but I like because it's super classic and really nice how they demonstrate how components of the microbiota, elements of the microbiota, not the Clostridium from the microbiota, not the difficile that is very harmful, right? you can see that oral inoculation during the first time period of life, and then the animals were subjected to this model of colitis with DSS, right? The experimental colitis at day 0. Look what happened with the, for example, the FOXP3, when the animals were fed with a probiotic, right? We can say a component of the microbiota, the Tregs are increased in the colon. However, and when they subject to colitis, the disease score is reduced compared to those not receiving this probiotic. What happened with the colon length is kept in balance compared to the counterpart. And the histological features are different as well. So what we see here, that the components of the microbiota, for example, Clostridium sp, Bacteroides fragilis, or some proteins from Bifidobacterium. I'm not a microbiologist, but this is super interesting to see that different components of the microbiota, they are important to shape different cells of the adaptive and innate immunity. But in terms of innate immunity, adaptive immunity, that is relevant to the effector functions long-term, for example, plasma cells producing IgA that is super important in the mucosa, the microbiota is relevant, and to promote the isotype switch to IgA and also the secretion of the immunoglobulin. For example, in colitis, that is inflammatory, chronic inflammatory condition, there's the production of IgA is quite reduced. FOXP3 is also absent. We saw in animal models, and this represents what happened in humans. Sorry. So in terms of innate immunity, there's a nice paper that also demonstrates that toll-like receptors that are important in sensing components of the microbiota in the production of IL-10. And they did adoptive transfer dendritic cells and T cells to wild type and deficient animals, and they found that TLR2 is the main sensor, and of course, if they use deficient animal cells coming from NYD88 deficient animals, they found exactly the same decreased production of IL-10. So microbiota is important. So dysbiosis is the alteration of our microbiota, and is nowadays associated with a lot of pathogenic conditions in the humans, not only in the gut, but also in other organs. So there's a balance between the good and the bad microbiota, and this population, cell population, immune cell population in the gut have an impact in other tissues, like this was nicely reviewed in this paper a few years ago, related to arthritis, metabolic diseases, allergic inflammation, another gut-brain axis also is involved in this system. And the gut produces some molecules that are important in shaping the function of immune cells, like short-chain fatty acid. Have you heard of butyrate, acetate, and propionate? Are metabolic products from dietary fiber digestion by the microbiota, that if you see a healthy colon here, you have a nice epithelium layer and anti-inflammatory cells or cells monitoring the tissue. However, in inflammatory condition or even in cancer, the proportion of short-chain fatty acids is reduced. We don't know if what is the chicken or the egg, no? If it's because the dietary fiber is reduced, or there's a lower fermentation because we have a pathogenic microbiota. The result is that we have a very low proportion of short-chain fatty acid, leading to an inflammatory condition. And that's why, because one of these components, the butyrate, that actually there are some patents of sodium butyrate in capsules, but it's much efficient, the one locally produced by digestion of dietary fibers. They can shape the production of anti-inflammatory cytokines like IL-10 by macrophages and dendritic cells that are resident in the mucosa, producing retinoic acid, and that they can shape, as you say in this diagram, the proliferation and differentiation into Treg cells, and reduce the activity of Th17. So there's several signaling pathways activated by short-chain fatty acids. Some of them are relevant for the proliferation and healing of the epithelial enterocytes or the enterocytes epithelial mucosa, but also they can activate histone deacetylases, promoting, blocking some transcription factors involved in inflammation. And the gut microbiota, we can skip this part because I want to show that fecal metabolites, not only short-chain fatty acids, are important biomarkers, not only of IBD, but also for diagnostic markers. You can see that some sphingolipids are increased in inflammatory bowel disease. This is a recent paper from our group, and IBD, the levels of sphingosine, for example, and they correlate with calprotectin, that you know is a marker, is alarming, but is detected in the stool and is a biomarker of inflammation in these diseases. And here I can show you that there's a metabolic signature that is completely different from healthy individuals. And you can do this, and was done in colorectal cancer and other nowadays cancer conditions. Like if these are the black are the, sorry, the controls are the dark, sorry, they are the black dots here, are completely separated to the inflamed tissue, and you can see if there's dysbiosis or not. Look, the dysbiosis is almost overlaying the inflammatory patients, right? The ones that have IBD. And here you see which are the metabolites that pop up in this inflammatory volcano plot. So there's virtually a interaction between the diet, the immune response, and the microbiota. That's what I want to highlight with these results. And beyond butyrate, there are important other components that are taken from starch, for example, using amylases in the salivary or on the pancreatic, that they can promote some carbohydrates that are substrates for the microbiota. So it's super important which are the nutrients and the diet that the patient is taking. And nowadays, I give you a paper that examples which are the components in the diet. If you are interested, you can read that also the therapy response. I can send you, Betty, and you please share to the students. I think I forgot to upload that paper, that the diet shape and contribute to the therapy response in different tumor conditions. So I will send you this paper that was published recently. So this is a paper recently published that I collaborate with this group, that another component that is important in the cholesterol metabolism, the LXR receptor, and links intestinal regeneration and tumorogenesis. And we demonstrate that in colonic organoids that is a very interesting model to see the epithelial proliferation, how cholesterol can promote the proliferation of epithelial cells, and when it's dependent on the, is not rescue, when this epithelial cells lacks of this receptor. How does it work? Sorry. This receptor is important in the tissue regeneration, and there are some factors or agonists that activate this receptor and promotes homeostatic molecules like RF to, this is a transcription factor that can promote the tissue regeneration, right? However, in tumorogenesis, it was shown that this pathway is also important, how anti-tumor CD8 or plasma cells can be affected in the tumorogenesis. And briefly, sorry, I want to show you, there are several pictures that in animal models subjected to colitis, as we saw until 70 days, almost 2 months, the absence of this receptor can promote the reduction of tumors. And they saw that the supplementation of in the diet of this agonist of the LXR receptor can affect the presence or not of tumors. I encourage you to read this article, because I think we have to wrap up the and in patients, this is super interesting, that we found that this enzyme that is key in the regeneration process and the lipid metabolism is increased in, is reduced, sorry, in colorectal cancer patients, and you can see here, and associated to mimentin is quite dramatic, the difference between healthy colon and colorectal cancer. And let me see if there's something else I would like to, well, this I will, is also to strengthen the importance of the dysbiosis in the tumor microenvironment. Sorry, and this, how the changes in, that's what I wanted to share with you, how this checkpoint blockade inhibitor are important when advanced melanoma patients are treated, and they associate with the gut microbiome. So also there's another study that associate how the diet, in this case they talk like Mediterranean diet, are important in beneficial for the response in advanced melanoma. And how different immune phenotypic phenotypes are linked to tumorogenesis and metastasis, and using the same rationale as we saw for the epithelial compartment, you can do exactly the same of differentiate immune feature of colorectal cancer compared to healthy individuals with or without liver metastasis. It's interesting because they found in this paper different T cell cluster, macrophage and monocyte cluster, and dendritic cells related to one condition or the other. And some other topic that I wanted to highlight is the importance of exhaustive CD8 positive cells, that these occur when they are constantly or repeatedly exposed to the antigen. They are not, it's a theory that they can be a reason that malignancy attributed to TCR dependency is when these cells become exhausted after repetitive exposure to an antigen. However, this theory is in debate. That's the expression, because these cells are hard to find in vivo. You can generate in vitro. However, it's quite difficult to exactly recapitulate the phenotype in vivo. And this is a result of these subsets in patients with liver metastasis. You can see that there are different cluster of cells that are related. So nowadays, you can, this kind of studies, what I want to highlight, they are more complex. That is not only a tumor-associated macrophage or a tumor-associated fibroblast important. It's the combination of all these cells, and it can be not the same in patients with the same pathology. So I think I will complete now, Betty, with this, and I share the class to you. I am happy to answer some questions if you have some. You can see that I have a bee here, and the bees work all together, and I think it's important that it's something that we have to always remember, that bees and research, we work all together to build something important and something big, like publications and novel data, and also discussion. So I'm happy to answer some questions from you.
Student, do you have any question?
Thank you, Professor, for such an insightful session. I have a few questions. So the first one is, I'm not sure if I didn't catch about the cause of dysbiosis in the guts, for example. So I wonder what causes the dysbiosis in guts. Is it because the cancer progression or other causes?
Thank you for your question. Who was the person? Because I couldn't see the name.
My name is Anuat Kanach. Thank you.
Thank you for your question. Well, the dysbiosis is a quite important topic nowadays, and I am working in this department of gastroenterology, and there are some researchers working in associating the dysbiosis and response to therapy, even change in diet. There's a protocol here, super interesting, that patients with IBD, they want to do now in melanoma, that if they get a root of chicory that is rich in inulin or other carbohydrates, they can improve the inflammatory score and the progress of the disease and the therapy response. However, it's hard to tell if it's the tumor promoting the dysbiosis, or if the dysbiosis affects the degeneration of the tumor. What is clear is that a healthy diet contributes to a better microbiota, and also to better prognosis of therapy, for example. But it's hard to tell. In the case of animal models, you can say that if you feed with the how you call, they call canteen diet, that if they give like high in sugar, in lipids, or refined sugars mostly, the appearance of polyps and adenoma is increased. For example, there are some animal models telling that. However, as we saw, the good microbiota can process or metabolize the dietary fiber. So it's a combination. You have to have a good diet and also a good microbiota. So nowadays, I didn't show, and here in the Netherlands, in many countries, the fecal transplant, have you heard of that therapeutic option, like fecal transplant? The person that asked the question? Yes.
I've heard of that, yes.
Well, you've heard that it's mainly used in patients with Crohn's disease, that they have Clostridium difficile infection, not the Clostridium sp that I showed that is part of the microbiota, but the difficile. They see a very good effect. But now they are here in some in Netherlands and other European countries, they are using not only for IBD, but also for other autoimmune diseases and also some autistic disorders that are quite, but I'm not familiar if there's some evidence in or results in tumor or cancer therapy response, because also some of the therapeutic options for cancer treatment, they can promote colitis. Maybe you've heard of that. However, the presence of colitis in the therapy response, for example, for melanoma or other conditions, is a good prognostic marker. So those patients, after the therapy, developing colitis, is a good mark of therapy response, because if they have inflammation, it means that the immune system is responding and can be effective to eradicate the aberrant cells. You know, it's a double-edged sword, but I think you can prevent, or once it's installed the disease, you can shape the microbiota with a good diet, and probably the therapies, any therapy will be more effective. Or maybe there's no, or change the dose of the therapy or the strength. But that's an oncologist's point of view, right? I don't know if some of you are oncologists or have some experience. No?
So the point here is, so the causation, although the causation of between the microbiota and like the disease is not clear yet, but many evidence suggests that promoting healthy diet can promote healthy gut microbiota and also promote better disease prognosis, right?
And the therapy response also.
Okay.
If you need, maybe you contact me or Professor Petty, and there are some papers in the literature, but I think one of the ones I put at the end of the class are related to that, to the checkpoint inhibitors, for example, how the diet and the restorage of the microbiota impact on that, and is beneficial. So no processed food.
Thank you.
So it's quite hard nowadays, right? But I was 2 weeks ago in the European conference of the Crohn's and Colitis Organization. So there were not much in colorectal cancer, but they showed that how in countries that were no incidence or prevalence of inflammatory bowel disease now is a big problem. For example, in India, and this is they associate to change in the diet because the traditional diet and food is changed, and probably you can see also in Thailand that there's a change in the. I remember your colleague Betty, this professor that was a surgeon in colorectal cancer, like he was in Mayo Clinic. What was this? Remember we have a meeting with your colleague from the hospital. He was a surgeon in colorectal cancer. And he
Yes, we have a collaboration with the surgeon with colorectal cancer. But right now he has some administrative work. So the work for the colorectal has reduced.
Yeah, but something that he mentioned, this was a colleague of Professor Petty, that he was in the US doing his postdoc, and when he came back to Thailand, I was lucky. I was in that meeting that he mentioned that the incidence increased quite a lot in the last 20 years compared when he was a medical student, and then he saw this dramatic change, and probably it's related. I don't know what you think, but the diet can be important. The change in the diet and the kind of sugars and the diet in the whole population. But it still is something to evaluate in more detail. Any other question, comment?
Can I ask a question?
Sure, yes. What's your name?
Yes. Diana.
Diana?
Yes. I'd like to ask about the macrophage in terms of its innate immunity, in terms of its role, and in the adaptive immunity in the cancer context. So when we look into the cancer, how we can differentiate in terms of its innate role or adaptive role? Can we say it is solely adaptive role? That I'm not familiar with that. So can you? Yeah.
Let me, if I understand correctly, the macrophages are innate immune cells. That's something that they are only innate immune cells. However, they can be affected by the adaptive immune cells. For example, a T helper, the CD4 T cell, for example. You know why it's called a T helper cell? Because it helps other cells. It can help a macrophage, can help a B cell, it can help even a cytotoxic T cell through the production of cytokines. So the CD4 T cells can change the phenotype of a macrophage from N1 macrophage that they are expressing CD86 like and secreting Th1-like cytokines like interferon, TNF, IL-6. But if you expose this macrophage to a Th2 helper T cell, remember that they produce IL-4, IL-10, immediately this macrophage, you can do it in vitro, they can change to an M2 macrophage. That is the most close macrophage to a tumor-associated macrophage. And they have have no phagocytic activity, they are not inflammatory cells. So this T cell conversation with the macrophage that is an innate cell can affect their phenotype and their function dramatically and make become a cell that is promoting and supporting the tumor microenvironment. So I don't know if your question is related to that, Diana?
Yes.
Are you working in macrophages?
Yes, I'm working there. So can we expect the macrophage in like the T cell, whether it can be polarized to kill the tumor cell? Can we like which way that we can target that?
Yeah, I think it's a good question. You can manipulate because they are quite plastic cells. If you, I didn't show that, but for example, if you have immune cells infiltrating the tumor, is a good prognostic marker, even macrophages or T cells. If they are in the periphery, this is not a good, histologically speaking, or if you talk to a pathologist, these are scores of prognostic marker or prognostic of the patient according where are the immune cells distributed. So if you have a macrophage that is M2 or tumor-associated macrophage, for example, with the diet, you can change some signaling pathway, the production of some metabolites that are important in the signaling pathways of macrophages or T cells, or even though you can for example, checkpoint inhibitors, or what we were seeing, some there are some also some cellular therapy. I think maybe Diana or your colleagues heard of cell therapy that, for example, in melanoma is extensively used. I have a colleague that he's in Karolinska, and also they apply in South America, and probably in Thailand, I'm not sure, but you can take mononuclear cells, myeloid cells from patients, for example, monocytes, and train in vitro to become dendritic cells that are effector dendritic cells. So then you can expose, train these dendritic cells or monocyte differentiated into dendritic cells, right, into a cocktail of cytokines, and exposed to tumor antigens, for example, melanoma. And then you re-administer to the patient these dendritic cells, and they've seen quite positive effects. These are innate immune cells, but they, as you remember, the dendritic cells, they can present an antigen to T cells. And if you have T cells that are more effector T cells, more CD4 effector T cells, they can, at the end, modify the environment of this tumor microenvironment and polarize M2 to M1 if they which are the main cytokines of a Th1 cell, for example, interferon gamma, TNF alpha, and these are perfect cytokines that they can switch M2 to M1, for example. So this is like a therapy in a therapeutic point of view. But you can shape the M2 phenotype into M2 and M1, if that's your question.
Yes. And for the cytokine choices, like there are a lot of the cytokines that can polarize that phenotype from M1.
I would say, would you like to do in vitro, for example, or for a patient?
For example, yeah, in vitro. So
in vitro, you can use, for example, a monocyte. You can differentiate with well, if you are not using in a patient with LPS and interferon gamma, and there are protocols. I can share with you, if you like, some protocols to differentiate M2 into M1 or M0 into M1. So you can expose to pro-inflammatory, like a pump, for example, in some, depending on which kind of M1 you want to recapitulate. If you like to F1 from the mucosa, use LPS or flagellin plus interferon, so you can differentiate into an M1. And then you can see not only surface markers, but also soluble markers secreted or produced by these cells.
Thank you very much.
You're welcome.
Okay, Marcella, I would like to say thank you very much for your very informative lecture, and you provide the idea and answer the question from the student, and we hope that you will give us the lecture for
Of course, yeah, I
next
send you
year.
in a PDF because it's a little heavy, but I send you in a PDF file. I send to you and you can distribute to the students. No
Yeah.
problem.
Yeah,
With
yeah,
pleasure.
yeah.
Okay. All right.
Okay.
Well, any question you can contact if there's any
Yeah.
experimental also approach you want to discuss. Welcome. Okay.
Okay.
Thank you so much for your.
Okay. Thank you so much.
Thank you very much for the invitation. I'm looking forward to seeing you soon. And good
Yeah.
luck with your projects and thesis and the course. Good day. Bye-bye.
Okay. Goodbye.
Bye. Bye-bye. Thank you. Miss you.
Thank you.